Hello, my name is Nicole Lynch, and this is Gregor Mendel's discoveries and their influence on today's food and medicine. Today, I'm not going to share my personal opinions about GMOs. There is, however, a lot of stigma around GMOs. Are they safe for human consumption? Are GMOs cost me more money? Why genetically modify something in the first place? I'm going to start from the beginning with the father of modern genetics, Gregor Mendel. Mendel was born in 1822 in Austria. While he was raised on a farm and expected to become a farmer, he instead joined the priesthood and became ordained in 1847. Once he was ordained, Mendel enjoyed substitute teaching. He did, however, need to obtain a teaching license. He unfortunately failed his exams to become a teacher. This led to Mendel enrolling in the University of Vienna to expand his knowledge of physics and mathematics, the two subjects in which he had been lacking. Mendel began experimenting with pea plants in 1856. After breeding thousands of plants in his relatively small garden, he chose 22 true breeding plants that were self-fertilized. True breeding implies that the characteristic present in the parent generation will continue throughout each subsequent generation. He then decided to cross true breeding plants in the hopes of hybridization. His experiments with hybridization led him to the discovery of inheritance laws. Upon completing his pea plant experiment in 1865, Mendel published his work titled Experiments in Plant Hybridization. In his paper, he detailed his methods, data, and even his thoughts on the reproductive cells of hybridized pea plants. His work led to even more discoveries in inheritance patterns. This was Mendel's last published work in inheritance. He then died in 1884 due to kidney failure. Near his death, Mendel was not the only scientist discovering new and exciting things about inheritance. In 1880, Hugo de Vries began experimenting with plant hybridization and published his work in 1900, not knowing that Mendel had beat him to the punch. Even after finding out about Mendel's work, he did not acknowledge them until the second publishing of his work in French. In 1892, Carl Correns began experimenting with trait inheritance in plants, but when he published his work in 1900, he became aware of Mendel's work and titled his paper, G. Mendel's Law Concerning the Behavior of the Progeny of Racial Hybrids. In 1898, Erich von Schermark began breeding pea plants. He was also aware of Mendel's work, but was worried his paper published in 1900 would not be taken seriously due to his inexperience. Everything Mendel discovered was confirmed by these three men. De Vries, however, felt that it was unfair that Mendel be named the father of modern genetics. Once again, Mendel's works were being recognized years later, this time by William Bateson and Reginald Punnett. These two were experimenting with sweet pea crosses when they discovered some unexpected inheritance patterns with their mutant pea plants. Bateson and Punnett discovered epistasis and complementation. Epistasis is when one gene is able to mask the phenotype of a different gene, and complementation is when two mutant parents can produce a wild type or phenotypically normal offspring. In 1902, Bateson published a paper, Mendel's Principles of Heredity, in defense of Mendel's work. Years later, Russian scientist Andrei Nikolaevich Belozersky discovered that plants contain DNA. Up until this point, it was thought only animal organisms had DNA. When experimenting with a horse chestnut, Belozersky was able to separate the thymine from the nucleic acid, confirming that the plant did in fact have DNA. This occurrence of DNA and RNA proved that microorganisms have DNA specificity. In 1957, with their knowledge of black operons, Arthur Pardee, Francois Jacob, and Jacques Monod conducted the Pajama experiments. These three hypothesized that in order for enzymes to be produced, something within the bacteria must need to be induced. When performing experiments on the bacteria E. coli, their findings did not support their hypothesis, but rather supported the theory that genes within the E. coli bacteria controlled enzyme production. They published their findings in 1959, and the Pajama experiments led to the uncovering of how genes in any other species yield enzymes. Stanley Cohen had previous knowledge of plasmids, and Herbert Boyer had studied restriction endonucleases. After meeting in 1972 at a conference, they decided to join forces. The Cohen-Boyer team at the Stanford University cut open the plasmid loop of a bacterial species, inserted the gene of a different bacterial species into that loop, and then closed the loop. This creation of a recombinant DNA was then successfully inserted into a bacteria, which proved that the DNA would be used by the new host. This was the first genetically modified organism. After the recombinant DNA experiment was a success, Paul Berg suggested that a conference of bioscientists, lawyers, and doctors meet to discuss the possible dangers of recombinant DNA. It was decided that because the DNA would not be able to survive outside of the lab without a human host, it would pose no danger, and genetically modified DNA could be used safely. Because the Asilomar conference was done out of concern from the bioscientists, This helped to gain the public's trust of genetically modified DNA. Now that genetically modified organisms were deemed safe, microbiologist Ananda Chakrabarty wanted a patent for his genetically engineered bacteria that were capable of breaking down crude oil. His patent was rejected on the grounds that living things are not patentable. Chakrabarty appealed his case to the Supreme Court, and a ruling of 5-4 agreed that because the bacteria was not discovered naturally and that it was genetically engineered, that it could be patented. Chakrabarty's bacteria was the first patented GMO. Not long after, in 1982, the first genetically modified drug was approved by the FDA. Eli Lilly and Company had successfully injected a diabetic boy with the insulin of a dog in 1922. The company then decided to manufacture a biosynthetic human insulin. The new insulin would show that there was little to no difference between it and the normal insulin produced by a human. Eli Lilly and Company worked closely with the FDA to manufacture this drug. It is because of the work done at the Similar Conference that drug was deemed safe for mass consumption. After FDA approval of a genetically modified drug, companies began to use similar technologies to create the perfect produce. Specifically, Calgene, a Californian company, decided to create the perfect tomato, the flavor saver. They reverse engineered the enzyme responsible for softening the fruit. This allowed the flavor saver tomatoes to remain ripened, but not soft during distribution to grocery stores. This was the first genetically engineered crop to be widely commercialized. Unfortunately, the flavor saver tomato fell flat in the late 90s due to high production and distribution costs. At the time, this technology was still relatively new and pricey. The flavor saver tomato walked so the GMO produce of today could run. Since the 90s, the production cost of GMO produce has reduced drastically. As of now, the technology is rather old, so GMO crops have been in rotation for years, allowing farmers to grow the produce at much lower cost to them. GMO produce is now resistant to insects, disease, and herbicides. Without extra costs bogging down the farmers, GMO produce is extremely affordable for the consumer. This way of farming has also gained popularity outside of the US, which has allowed farmers in other countries to earn more money without the pesticide exposure. As of now, there is no evidence that GMOs can cause health problems. After learning a bit more about GMOs and how they came about, I'm a bit curious as to whether or not we are better now with GMOs, or would we have been better without them? As I said before, I'm not here to state my opinion, but only provide factual information on the subject. I will let you decide for yourself if you are pro-GMO or no GMO. Thank you so much for listening to my presentation. I hope you learned something new.